Welcome back, friends. In this video, we'll be talking about Listeria species, especially about Listeria monocytogenes. And this Listeria monocytogenes is a part of a gram-positive rod. So definitely these are free-living, and this is a rod. Okay, rod-shaped bacteria. So the first property you can write, this is a gram-positive rods. Okay. And what we can see about Listeria monocytogenes is that they stain very darkly. So the very dark staining. So they take very dark staining here. Okay. And they are kind of short rods. So they are short rods, not very large. They're short rods. That means if I draw their structure, it will look something like that. So it can be short rod like this. So uh, you may think that they are spherical, but they are not spherical. They are short rod like that. And usually they can sometimes find as diplobacillus like this, two rods all together. This kind of structure is called diplobacillus. So let me write it here. So you can find them as diplobacillus, diplobacillus, or what we can also see that they are attached with one another to get a chain like structure like this. Okay, so we can get short chains like that. Okay, these things can be fine. Okay, so and another thing is that they are intracellular kind of parasites. So they are intracellular parasite. That means they never comes out of the cell to infect. They only can infect cells when they are inside. So parasite. So they are intracellular parasite in nature, and they are catalase positive, and they are having a movement. So they are having a movement, and the movement is here. So let me talk as tumbling movement can be seen for Listeria. So they are having that kind of flagella, and they can have that tumbling movement. Okay, they grow facultatively on various enriched kind of media. So we can see these are the major properties about the Listeria species. And among the Listeria monocytogenes, we have seen all these things. So under the microscope, we can see them as a diplobacillus or short chains like that. Okay, sometimes we can also see them as a separate bacteria, but very rare cases. Okay, and Listeria monocytogenes is clinically significant because they can be found. So let me talk about it. This is very, very important. They can be found uh, almost. So they can be found in milk products or dairy products. As well as we can also find them in ground meat. That is making them very, very significant because in dairy products we can get almost 2 to 3% of this Listeria contamination, and in ground meat we can see up to 20 to 30% of the contamination, which is a very high level of contamination by this Listeria monocytogenes or Listeria species. Now, among all these different Listeria species, this Listeria monocytogenes is found to cause any kind of disease in human being, and the disease most of the time is prevented by our own immune system. So this is not that much threatening for us, but this kind of disease is life threatening for pregnant women. So let me write it here. It is the disease is dangerous for one is a pregnant woman. So let me write here. So one is pregnant woman, and they can be dangerous for immunocompromised people. Immunocompromised, right? Compromised person. Okay. Otherwise, this Listeria infection can easily be treated by our own immune system. It can be found. The agent Listeria monocytogenes can be found and can be killed. But in pregnant women, it can be dangerous because in pregnant women, before the delivery and during and after the delivery, also newborn can also catch this kind of infection. So, from the pregnant woman, newborn baby can also catch this infection. right? So that's a serious concern, right? Because newborn baby is not having the immune system that much strong at that time, so infection thrives. Uh, we don't have any choice, right? But otherwise, this is very basic, very simple kind, so we don't need to bother about much. Okay, and also this kind of disease actually they can cause any kind of what you can say, swelling of glands. These are the different symptoms: swelling of glands, and in pregnant women it can cause several kind of difficulties, and during the birth it can cause disease. It can infect in the vaginal tract, so during the movement of newborn baby through that, they can catch disease. So this is in a whole about Listeria monocytogenes, and the very important process or uniqueness of Listeria monocytogenes is its incredible ability to be present intracellularly or to be transferred intracellularly, right? So we can see the intracellular, so let me talk about it, intracellular movement, intracellular movement from one cell to another cell without being exposed to the extracellular surface. So that is very unique. That is really unique to these Listeria monocytogenes, and we are going to see this effect in future slides. Okay, so that's it, and I hope that's helpful. Thank you. Welcome back, friends. We have already seen the introductory video about the Listeria monocytogenes. Now, in this video, we'll be talking about the infection caused by Listeria monocytogenes or the clinical significance of this. So, we'll be talking about the infection caused by this Listeria monocytogenes bacteria. Now, this infection that we are going to see caused by Listeria monocytogenes is called, as it is called, listeriosis or listeriosis. So let me listeriosis. Listeriosis. Okay, the clinical term is listeriosis. Okay. Now the listeriosis is a variety of focal lesions, and they are less frequently seen now, such as granulomous skin lesions. So it's a kind of focal lesion. So let me write. This is a kind of lesions. Sorry, it's a kind of lesions that we're going to see. Lesions, lesions like granulomatous lesions. So let me write it: granulomatous, granulomatous or granulomatous skin lesion, and also we can see other types of lesions. Okay, so you can call it as focal lesions. Anyways, so you can find this kind of lesions formed. Okay, and also it is much more common in pregnant women. We have already talked about that. It is very much common in pregnant women, as well as it is very much common in those individuals which are who are immunocompromised. Okay, and actually in this case of pregnant women and in many women, what we can see that vaginal colonization of Listeria monocytogenes. Now this vaginal colonization can also be seen. So vaginal colonization can be seen. Now this vaginal colonization can also lead to the problem of this pregnant woman, as well as it lead to supply this particular bacterial infection to newborn. Okay, to newborn. So let me write newborn, and it finally triggers the change in newborn, and finally results in meningitis. Meningitis in newborn. So this is a serious concern for the newborn babies, right? Or sometimes it can also infect a fetus, and also so this is the third round. So this is the first kind of infection. So let me write, this is the first kind of focal lesions. Second kind is the vaginal colonization due to the newborn infection, and the last kind that we're going to talk about, and this kind of infection is completely different. It is infecting, it is also infecting fetus. As it is infecting fetus, as a result of this fetus infection, it will lead to abortion. So in this case, as you can see, in normal individuals, it won't lead to that kind of dangerous effects. But in pregnant women as well as in newborn, they are providing this meningitis, abortion, and all these things, which are bad, which are really dangerous things, right? So that is one reason. And another reason is the immunocompromised people. The second part is for the immunocompromised, because the immunocompromised people are having very weak immunity. So as they are having very weak immunity, as a result of that, they can catch this disease. Pretty fairly, right? So that's why this is another major concern of our interest. So one is a pregnant woman, and the second thing is for the immunocompromised peoples. Okay, so this is all about the infection, and I hope that's helpful. Thank you. Welcome back, friends. In this video, we'll be talking about the Listeria pathogenesis, right? So it's about the pathogenesis. Pathogenesis. Now, what we know about the pathogenesis for all microorganisms, as I've told you always, that pathogenesis consists of two different strategies. First strategy is to escape immune system. So let me write the two strategies again and again. I've written this so many times right now. So first strategy is to escape immune system, right? Escape immunity or escape immune system, right? And the second process or second way of doing it is to the destruction of host cell, right? So these are the two important strategies for pathogenesis of all bacterial cells, virtually all bacterial cells that we're talking about. Now, in this case of Listeria infection, they are managed to take one strategy only. So only one strategy. So it's a master strategy. So this one strategy will fulfill both of these criteria on the same frame. So it will fulfill the criteria of escaping immune system as well as destroying the host cell. Both of them with one master strategy. And the strategy that we are talking about here, and normally what we know is that there are some immune system cells like macrophages, which are called the phagocytic cells, right? So we know these macrophages, which are phagocytic cells, and also neutrophils and all these. So neutrophils and all these cells are there. They are immune system cells. They can find this bacteria, they can engulf them, kill them, right? Now they need to escape all these cells, and they also need to destroy the host cell. They need to colonize, right? That means they need to colonize. So to do all these things at the same frame, what they are doing, they are choosing this immune system cells. They are choosing these cells like macrophage, neutrophils. They choose these cells as their habitat, right? So they are attacking these type of cells. So this is the master strategy to attack this macrophage or neutrophil like these immune system cells. So they are attacking immune cells, right? Immune cells attack is seen now. When you look at the immune cell attack, now in this case, so we are having macrophages. So let's look at the structure. Let's draw the structure. Let's say this is the Listeria, small rod Listeria. This is the Listeria. It is engulfed by a macrophage. So let me draw a macrophage for you. This is the macrophage. It is now engulfing this particular bacteria here. So it is engulfing. After the engulfment of the bacteria, what we can see is that we are having this macrophages, and inside this macrophage, what we are having this vesicle inside which we are having this Listeria bacteria like that. Now, once it is engulfed inside, this is called the phagosome, because this particular process is called phagocytosis. And the vesicle inside which the bacteria is in now is called the phagosome. So this is basic immunology concept. Now it is phagosome. Now after the engulfment, what is it is doing that they will fuse this phagosome with a lysosome, which is an enzyme-filled sac inside the cell. So let's say here inside the cell, this is enzyme-filled sac. So this is literally is lysosome. lysosome. It is filled with hydrolytic enzymes. So whenever you heard the name of lysosome, that means it is filled with hydrolytic enzymes. Okay. Now they are present inside lysosome. So after the engulfment via phagosome, the phagosome will fuse with lysosome. Now after the fusion of phagosome with lysosome, so let me draw this macrophage again here. So here it is a macrophage, and after the fusion, what we get? So these two things fuse with each other. So here it is, and here we go to lysosome. The fusion occurs at this particular case. After the fusion, this bacteria or Listeria is inside this phagosome. Now, as it is inserted into the phagolysosome, this Listeria, normally what happens in normal case of infections, our immune system cells depict it as a threat. It will engulf it, fuse with the lysosome. As it is fusing it with lysosome, lysosomal enzymes will start to destroy this bacterial cell right now, right? But in this case, what we can see is this is called the phagolysosome. Let me write it. It is phagolysosome. So in normal case, after the engulfment of inside the lysosome, there is no way to escape. But in case of this Listeria infection, they start to secrete a chemical mediator called Listeriolysin O. It is called Listeriolysin O. So let me write it is called Listeriolysin. Listeriolysin O. So as the term suggests, it is a lytic kind of enzyme because lytic kind of enzyme. So this listeriolysin O enzyme will degrade this phagolysosome. So as it is now providing this, it will start to degrade this phagolysosome. As it is degrading phagolysosome, what it will result? So let me scroll a little bit down. So here, as a result of that, so let's say this is the macrophage here. Now inside that, it degrades it. It degrades this phagolysosome. So it's a degraded component of phagolysosome. And what it get is that it is now become free bacteria. Now as it is becoming free, now the second step of the pathogenesis will occur. So the degradation is done via Listeriolysin O. After that, the Listeria start to multiply inside the cell. So few times later, it will have a lot or many number of Listeria inside this phagocytic cell, inside this macrophage. Now, as we are having more and more Listeria, now they start to accumulate actin filament. Now, we all know that actin filament is a very important component of a cytoskeleton of eukaryotic cells, as you can see, as we know. Now, they start to rearrange. They start to secrete some chemical mediator, some enzyme, which start to rearrange these actin filaments. Okay, so let us take this particular thing. So let us take this one among all of them. Let us take this one as an example. Now let's follow its journey here. Okay, so let's say in the future place, what we are going to see that yes, this is the macrophage is standing here like that, and inside the macrophage here it is our Listeria. Now this Listeria start to arrange those actin filaments. Now let's say these are the actin filaments. So actin filament assembly start to occur. Now as actin filament assembly start to occur, it start to form what a kind of comet-like structure. As you can see here, it is a kind of comet-like structure. Let us come here. It's a comet like structure. So it's a bacterial comet, right? Bacterial comet that is formed in this particular case. So comet like extension start to form. Now after the formation of this comet-like structure, this actin filament will start to build and it start to push this bacteria forward. Now as it is pushing the bacteria forward, this bacteria starts to move in forward direction. Let's say here it is again the macrophage. As it is pushing forward, it is going towards this. So previously it was at this place, now it is coming towards this cell membrane, and it is due to the pushing effect that is bring about by this comet structure here presented with this actin filament, right? So it is due to, so let me write, it is due to the actin assembly. It's due to the actin assembly. Okay, so it will push the bacteria forward, but now it will reach at the terminal of the cell membrane, and then it start to come out along with the cell membrane. So let us draw this picture now. So let's say here it is. This is the sorry, let us change the color here. This one is the macrophage. So this is the macrophage again. Now, as it is, as we can see that it start to push the cell membrane further due to the formation of this comet-like tail. So as a result of that, it is creating this kind of extra invasion-like structure, and with the help of this structure, it can attach with the future macrophage. So let's say this is another macrophage. Let's say this is another macrophage, and as it is holding this kind of structure, this is a large macrophage. This macrophage is again, this is another macrophage. Now, as the listeria is going out, this macrophage will start to engulf this one, right? So as it is moving, and then it is making this kind of pseudopod like extensions. Now this pseudopod extension will facilitate the movement of this particular listeria bacteria along with the cell membrane of, remember, along with the cell membrane of this macrophage, along with this macrophage, right? So after this, what will be the engulfment? After the engulfment, we can see some kind of structure like say, say this is the macrophage. It will look like this, and this new macrophage will look something like that. After the engulfment, let's say here it is. After the engulfment, what we can see, we are going to see uh, a vesicle like that made up with this, and a vesicle like that made up with this one, and inside what we are having our bacteria. So here it is the listeria. So this is the engulfment. After the engulfment, using pseudopod, right? So these are the sequential stages. These are the sequential stages of the occurrence. Now this macrophage will become free from the listeria. Not actually free, because there are other listeria that are present there, and they will start to secrete some proteins to destroy it, and also they will start migrating or moving, right? So these are the different mechanisms of the pathogenesis. As you can see from the beginning, engulfment, then utilization of the listeriolysin O to degrade this phagolysosome content, and then finally using actin assembly to produce this bacterial comet, which is helping them to arrange and create the pseudopod to escape from one macrophage to another macrophage. That's how they can transfer via intracellular movement. So as you can see here, this is via intracellular movement. So let me write, this is via intracellular movement. So in this whole movement procedure, there is no exposure of this bacteria extracellular region, in extracellular region, right? So it's an intracellular movement, complete intracellular movement that you can see. So in all these cases, in normal bacterial infection, what will occur, that one bacteria will escape to the extracellular junction, then it will move to the another cell. But in this case, it is the direct cell to cell entry, and that is the very uniqueness of this listeria infections. Okay, so that's the overall procedure of pathogenesis for listeria, and I hope that's helpful. Thank you. This is going to be a short video on the treatment of Listeria monocytogenes. So it's a treatment. It's on treatment of Listeria monocytogenes. Now the treatment for Listeria monocytogenes is pretty easy, because this Listeria monocytogenes infections most of the time is not that much severe, that can be easily treated using common antibiotics. Now the antibiotic that we can use is of the penicillin type or penicillin family. Now the typical type of antibiotic that you can use for the penicillin family is ampicillin. So you can use ampicillin in this case, right? Sorry, ampicillin in that case. And for the other type of medications, we can use trimethoprim. So let me write, we can use trimethoprim, as well as we can use sulfamethoxazole. So sulfamethoxazole. So we can use these antibiotics to treat Listeria monocytogenes, and they are very much effective, very effective against it. So very effective against this kind of infections. Okay, very effective against listeriosis. But the most important thing about the listeria infection is that before the treatment also, you need to be very careful about your food habit and your handling of the food, because these listeria infections mostly provided using the dairy contamination as well as meat contamination as well as ground meat contamination, right? So if you're using or if you're cooking ground meat, you should cook it properly. So proper cooking is required to eliminate this Listeria pathogen. As well as for the dairy contamination, you need to be very careful about choosing good brands and follow all the guidelines to use it. Okay, so if you follow this particular things and uh, then most of the time Listeria won't infect you. If it infects, your immune system is strong enough to go against it, so you don't need to worry. But if you are immunocompromised, I'm repeating, if you are immunocompromised, then you are having a risk for the Listeria. Actually, if you're immunocompromised, you are having risk for any kind of diseases, right? So it's a hell of a mess. And otherwise, also for pregnant women, you should follow some important guidelines of choosing these products, right? So that's it, and I hope that's helpful.